This is a first. Well, you all know the drill. You present your science that got you excited, and we talk about it and have a discussion about it to the best of our knowledge. There is no wrong questions. Thanks for the link in the chat. Everyone can follow. I'm going to put that over on YouTube as well. And today we'll see how we get on. If we take more than half an hour, I'm going to hand over to my lovely colleague Valentina, because I have a meeting that clashes today. But if we finish on time, then that will be fine. Right, let's make a start. Hervé, do you want to take it away?
Yes, so let me share my screen quick. Now, yes. Okay, so I wanted to present another paper when Stephanie, we saw a paper on Twitter, and but I decided to present another paper, which is about the same topic. So it's about harmonization. So this paper is about harmonization of large dataset of MRI. So the title is Harmonization of Large MRI Datasets for the Analysis of Brain Imaging Patterns throughout the life span. So everybody noticed we are in the era of population imaging, big data. And so that means to mix a lot of data together. So mixing different scanners, different samples to be able to conduct a very large analysis. So the problem when you do that is you have a problem of scanner effect. So you mix different scanners, different sequence. So you have problem with segmentation, for example, when you want to segment the T1 images. T1 image, you have a problem on the, for example, on the contrast between white matter and gray matter. You will not be able to segment the same image on different scanners because the contrast is not the same. So that is a kind of problem you have when you mix different data sets together. So people are trying to solve this issue by harmonizing the data sets. So that means removing. So there is different ways to do it. So you can do at the scanner level when you try to, you have your image out of the scanner and you try to change the contrast to match the different data sets. The other ways to do that at the statistical level, when you try to remove the mean and the variance of the different data sets. So this paper is more about this kind of strategy when you try to, at the statistical level, to match your data sets. So, and they took the example of looking at the age effect. So for that, so let's get through the paper. So it's about aging. It's about age-related effect and on structural MRI. So they selected 18. Let me make that bigger if I can. Yes. They took 18 studies with different age range. So it goes from, I think, 3 to 96. So a very large age range. And so, yes, it's 10,000 subjects and from different data sets. Okay, so here they want to look at the age effect on the different structure of the brain. So one interest of this paper, they took two strategies. The first is to, at the modeling part, they took, they didn't use a classical linear approach. So they use what they call GAM, so which is a generalized additive model, which allows to do nonlinear modeling. So nonlinear modeling, and in this case, it will be nonlinear modeling of age effect. And the second strategy they used is they took an algorithm called combat. So I don't know the acronym what that means, but we can find out. So the combat algorithm was derived from genetic studies. So it was made to correct for the different genetic arrays. When you have different, so you extract genetic data, you have problem when you mix them together, so you don't have the same arrays, so you need to correct for that. So so a paper did that, so using this strategy for MRI studies. So here they do the same thing. So two elements. So the combat algorithm and the GAM modeling. And what is cool about this study, they use a R package, which I used a lot, and I like a lot R because it's for, at least for the statistical, from the statistical point of view, it's, I think, very efficient. So let's go on what they did. So they selected, so they took all the subjects together, and they started to do estimation and to try to I'm trying to find a word, simulation. So they simulate different elements. So the first thing they wanted to test is if the GAM, so the generalized additive model, was better than the linear. So here you have, so I think they selected 3 sites of from the 18th, and they took the hippocampal volume, and they look at the age effect. So in blue, you have the additive, the GAM, so the generalized additive model. In red, the linear, and in green, the quadratic. So you can model. So linear, quadratic are kind of regular statistics. They are linear, and the GAM is non-linear. And so what you can see on these different data sets, first of all, when you look, so it's very important to see on the age range you are studying, the pattern can be completely different. So, for example, for the PNC, which is very developmental, you have kind of linear increase. And you see it doesn't change a lot to use a different statistical model. But if you look at more aging process, here it starts to change. So the linear is not so good, and the quadratic and GAM is kind of fitting well the data. And they did a comparison in this table, and just the conclusion is the GAM estimation of the fitting of the data is better with the GAM versus the linear and the quadratic. So in number, so this table show you that for the number of airwise, so maybe I didn't precise that. So the method they use is a kind of segmentation of different region of interest. So they derive 145 regions, and they computed the volume for each of these regions. So here they show that using a non-additive modeling, you fit. You have a better fit of the volume, mostly with the GAM algorithm. Okay, so that is good. So then, after that, they did other simulation. Though here they you have 2 sites. So let's say you have 2 sites, 3 sites, seat A and seat C, and you change the mean and the variance of the seat B and site B, and you want to see if your algorithm is behaving properly. So you do that. So they change the mean and stuff like that. So so here you have the conclusion of that. So here you can see the median age of site B. which is what they wanted to test. So in red, and the numbers are the estimation error. So the bigger it's worse. And here they change also the sample size. So they show that so if you are under 200 subjects, your estimation error is bad. And for the median age of the site B, if you change it, so I go back to the here. So they move it close to site B, site A, in the middle, kind of, and completely on the right, close to site C. So it's not a big deal, but the best is to have something in the middle between the two samples. So it's good to have a good overlap of the different data sets you are using. Okay, so here they played a little bit with the sample size again. So again, site B, the thing they wanted to simulate here was if you have a data set which is bigger than the other, is it bad or not? So here you have still the site B with 4 times bigger and the same size as the site A and site C. So what the conclusion of this simulation or this experiment is the bigger is the better. So it's better to include all the subjects in your analysis. So here again, the estimation error is smaller when you include all the subjects in it. Okay, so that was about the modeling. So here the combat algorithm. So here you have the raw data for the hippocampal volume, and after correction with the combat. So you can see that the mean and the variance are corrected for it. So here they took the hippocampal volume, which is working pretty well. I can show you in the supplemental they did other regions. For example, so here, so you have the amygdala. Okay, so you see that even for the total gray matter, so you see that there is a large variability in the total gray matter. And after correction, it's working well, but you have still differences. I think for some other regions you have no, only for the total gray matter. So it can vary based on the region. Okay, so combat is doing well. So now let's try to do the combat plus the GAM. So here, the way they tested it. So when now, I don't know if you are familiar with that. You have this kind of simulation of the age of the subjects. You can extrapolate the age of the subjects. It's called brain age. So you can simulate or you can predict the age of the subjects based on the data you have. So here they do that. So you have the different volumes of the LRIs, and you estimate the age of the subjects based on these volumes. So you have the real age of the subjects and the predicted age. So they look at that with the raw data. So here you have on one side you have the predicted age and the actual age. So it's fitting well, but you see it's kind of bumpy for some data sets. For the raw data, so with the combat plus only the linear modeling, you have a better fit. And with GAM and combat, you have the best fit. So the MAE, which is the estimation error, is smaller when you combine the two algorithms. Okay, so they were pretty happy. So after you have the real data when you have corrected for all the with combat, and when you are using the additive modeling. So, and you find kind of what you expect to find with this kind of large sample. So you have a decrease of total gray matter volume with a fast decrease due to the development during the first between 0 and 25, let's say. And after you have a slow decrease due to the aging effect. Total white matter is kind of classical. You have an increase kind of plateau, and the aging process takes over after 50, 60. So everything is fitting well for these regions, and here you can find also these third ventricle, the CSF is increasing. What else? The deep gray matter is also what you expect to find. So an increase during the adolescence and a decrease after all. And I think, yes, they did also, so they have a website for this study. Let's see if I have it, where you can get the results for your, for the region you are interested. So for, let's say, let's take something interesting. I don't know, the fusiform left. And you can, you can set some, so you can look at the data and see the estimation for example, here for the, so this is the left fusiform gyrus, and you have the decrease with age for the male and the female. And I think, yes, so no, it was cool. I have some questions. I had some questions about that. So if you had, so these data sets seems to be very, very clean. So I don't know with bumpy data sets what is the output of that. They discuss about also the geographical diversity, because the problem with when you combine data sets, the majority of these data sets are European, American, so you don't have a lot of Asian, Middle East, or even African data sets. And also, if you look more specifically to aging or developmental part, so here it's a very large age range. So if you focus on a very specific, a very narrow age range, is it different? So, but yeah, it was cool. So it's I think this paper is a case example. They wanted to test it on a regular age effect. So, and what they want to do is to look at the, for example, with disorder, psychiatric disorder or neurological disorders. I think that would be cool to do that. And also, I took this paper because there is a new one out. Let me show you if I can. And this is the last, so this is very recent, and here they use a longitudinal combat, because this study was about transversal, only transversal studies, and so the way to go now is to conduct longitudinal imaging analysis, and so they adapted the combat algorithm to deal with this kind of longitudinal data. And the end, I think I was not so long, so good.
Lovely, thank you very much. Are there any questions in the room? No one?
So Anna, can
yeah, sure, go for it.
If I understood well, they wanted to put together all these different sites, MRI scanners, because they think like the numbers, it's significant in this age estimation evaluation. And so like which were the main problems that they had in combining the different scanners. So the combined algorithms was able to harmonize everything, or did I understood well?
Yeah. So that was the idea. Yeah, this is the estimation. So yeah, as I told you, the estimation of age seems to behave more properly when you add the combat harmonization. So meaning the fitting, there is a better fit of the data. So better fit, and also the age prediction fit with the actual age, meaning you are closing with the real data when after correcting for this kind of noise and bias in the different data sets due to the scanner and segmentation errors.
And also it seems from this plot that at around the age of 25 is most noise, or maybe variability.
Yeah, I think they have issue with two data sets. So yeah, you have the two data. So it's and I think this is a younger data set. So let me show you where this sample. I think they have these different data sets which are for younger subjects. So the age, the variance, they have a large age range, and I think they have there is more variability in these samples. And I didn't dig it too much, but I think maybe there is more scanner issues in these data sets, or I didn't.
Might be also just fewer subjects if you compare to others on age range.
Yeah, because they have also the UK Biobank somewhere, but they selected only a few, not the full sample. They have only one, so I don't remember the number, but it's not the full UK Biobank. But I think it's that. So they have fewer subjects, and it's why you can see the bumpy, even here, not here, not so much here. But here it's only, yeah, you see it's not selecting, they are not in here. But if we go back here, yeah, here you can see these two samples here. They are kind of bumpy, and even after, it's kind of okay. But as I show you, I think where is it?
I'm just going to use that time to hand over to my lovely colleague Valentina, because I need to join the other meeting. But I left you with a couple of questions, so you keep the discussion going. And thank you very much for the paper, Evie.
You're welcome. Yeah, so you can see here they are more bumpy anyway. So you can correct for it, but I think when you don't correct for it, you have this kind of bumpy air, and it's back.
Okay, so the correction seems to work well.
Yeah. So this is the raw data, so it's why it's bumpy here, and after correction, it seems to disappear. So
okay, perfect. Thank you.
So I had a question. I'm not completely sure how they corrected the data. You probably have explained it, and I just had a lapse of attention. But
yeah.
It's like they are only correcting the volume, the statistical data of the volume, right? They are not doing anything on the images.
No.
All right.
No. This is a strategy they took.
No
so
problem.
you can play on the MRI side, but here they wanted to use only the volumes that you extract and to harmonize, which is very simple. You change the mean and the variance. You center your data, and all your data need. So it's why this. You want everything to be at 0 and with the same variance.
So they train their algorithm on some data and test it on other, or they train it and test it on the same, or
they did that also. So there is all so I don't know. They did different kind of validation of their algorithm. Trying different strategies. I don't know where I can find it, but yes. So 10 randomized repetition, and looking also at I cannot find it, but yeah, they try different validation strategy.
Okay, thank you.
And I guess that the idea is that the more information you use, the more precise the algorithm will be.
Yeah. You mean when they try different samples?
Yes.
Because I think the issue is when so when you mix different data sets, sometimes you have a very large data set which takes over. So you have, let's say, you have 10 subjects and you have a sample of 1,000 subjects. What is the advantage to take this small sample of 10? And I think they wanted to know if there was not a bias if you have a very large sample and you have small samples. And so the conclusion was that the estimation of age, but I think it's it really matters. It really depends on the age range and the range you are looking at. But on this study, looking at a large range where you have large patterns of age effect, maybe it's good to have the maximum subjects, maximum sample size. But yeah, here they tested here in this figure you have the side B, if you remember. They wanted to test if you have the full sample, which is 4 times bigger, 3 times bigger, 2 times bigger, and equal size. So when you have equal size, your estimation is not bad, but it's better to anyway. Even if you have unbalanced analysis, it's better to have all the subjects in the same analysis.
All right. Do you have any more questions? I can read the questions that you left in the chat. How is it to combine kids and adult brains to run a comparative analysis?
I think that is so here as I say in conclusion, it's in case example because it's true, and even here you see the classical pattern of development and aging, but the most interesting part will be to study even younger subjects, because here you have for the gray matter, let me show you. You have only the decrease, total gray matter. You have only the decrease and not the increase, which is make the modeling more difficult, because here you have only decrease, so you can model very precisely the decrease, but you don't have increase. For the deep gray matter, you start to modeling the increase and decrease. But I think it will be interesting to test a different age range to see how it behave on this very small age range, to see if it is able to describe properly the different trajectories, developmental and aging.
Yeah, it would be also interesting to compare the increase and decrease in white matter with cognitive data, for instance, to see correlations. And then Sophie, congrats with the website. She said the website is cool. And then she asked, any clinical use of these data? What can be the clinical use of this data? And then
so I think it's what we are missing here. It's they wanted to test it, so they use very stable framework. So here it's only healthy subjects. So I think it will be interesting to see if, because like Victor noticed, so the way of harmonizing is to correct for mean and variance. So saying that you expect the variance to have a kind of linear estimation. But looking at the pathologies can be more tricky in the way the variance itself, not even the mean, but the variance itself can be very different from comparing healthy to patients, for example. So that will be more difficult, I think, for this algorithm to extract. But even that, so it will be better to use this kind of strategy, but with some, I think you'll get more error in the modeling using neurological or psychiatric disorder sample.
And the final question is, what's the impact of small size between kids and adults?
Okay, so you asked me this question before. Now that was not the same question. So what do you mean? That is Stephanie's question?
Stephanie's question, so I'm going to interpret the question as, I guess, if they take into account the variability of the size of the brain structure.
I think they tested it in the way they wanted to know if changing the sample size was affecting their estimation, but I don't know if they specifically look at if it's true that you have a bias in the way you will have more adult subjects, because it's easier to scan them compared to kids, where it's more difficult to scan them. So you will have smaller, which is bad, because as I showed you, the estimation in adults can be kind of linear in a way, but in kids and adolescents you will have kind of very different trajectories, and even between subjects. So it's kind of tricky and more difficult, and you have less subjects to estimate this kind of modeling. So yes, I think the bias would be more on the kids and adolescents part and less on the adults.
Okay, thank you so much, Hervé. Any questions in the room? All right. So thank you so much, and see you next Monday for another Neuroccino with all of us. Thanks again, Hervé. Thanks to everybody in the room, and also everybody who's watching from YouTube. And see you.
Thank you. Bye.
Bye.
Bye.